I feel like we all have that one uncle or maybe a friend's grandfather.
Oh, yeah. The guy who smoked a pack a day for 50 years.
Right. The guy who smoked constantly, ate bacon for breakfast every morning, and somehow lived to be 95, completely cancer-free.
Yeah, it defies all logic.
He really does. And then, on the flip side, we hear these completely heartbreaking stories of people who have never touched a cigarette in their entire lives.
Right, people who exercise, eat perfectly.
Exactly. And they still end up sitting in a doctor's office getting a lung cancer diagnosis.
Huh.
From the outside, it just feels entirely random, like this terrifying invisible lottery.
It really does feel that way to most people.
But what if it isn't random at all? What if the difference between those two people is actually hiding in the microscopic, genetic switches of very specific individual cells in their lungs?
Well, that is exactly what we're going to get into today.
Exactly. So welcome to this deep dive. Today we are looking at a genuinely groundbreaking 2026 paper from Cell Genomics. It's titled Single-Cell eQTL Mapping Reveals Cell-Type-Specific Genetic Regulation in Lung Cancer.
Which is quite a mouthful.
It is. But our mission today is to uncover exactly how inherited genetic variations cause lung cancer, not just looking at the whole lung, but drilling all the way down to the individual cell level.
And I just have to say the sheer scale of this source material is genuinely breathtaking.
Yeah. How big are we talking?
Massive. The researchers behind this didn't just, look at a few random tissue samples and guess at what was happening. They built the largest single-cell eQTL atlas of normal human lung tissue to date.
Wow.
Yeah, we are talking about physically examining 562,255 individual cells.
500,000 cells. That's wild.
Right, and they categorized those into 17 distinct cell types, all taken from 222 Chinese donors. They essentially mapped the genetic dark matter of the lung, literally cell by microscopic cell.
Okay, let's unpack this because I want to make sure we are totally grounded in how they actually achieve this. The paper throws around some really dense terms like GWAS, EQTLs, and scRNA-seq.
It's a lot of alphabet soup, for sure.
It is. And I think to really grasp the magnitude of these findings, we need a solid handle on the tools they use to get there. So my understanding of a genome-wide association study is that it's basically like a giant treasure map.
That's a great way to think about it.
Right, because for decades, scientists have scanned the genomes of thousands of people to find these genetic risk areas for lung cancer.
Yeah.
And they found over 50 of these susceptibility loci, right?
I have, yeah. But that treasure map has a really massive flaw.
Right. It tells us where the risk is, but it rarely tells us what the risk is actually doing.
Exactly, because most of these genetic flags, they don't land directly on the parts of our DNA that actually code for proteins. They land in the non-coding regions.
Which geneticists usually call the dark matter of the genome, right?
Right. So, there is a genetic mutation associated with lung cancer at this very specific coordinate, but you have literally no idea which actual gene that mutation is influencing.
Which brings us to the EQTL, the expression quantitative trait locus.
Yes.
I always like to visualize the EQTL as a light switch. It's the wiring that connects that random spot in the dark matter to the actual gene it turns on or off.
That's perfect. It finally tells us what the mutation is actively regulating in the body.
But what makes this 2026 paper so revolutionary isn't just that they are looking at these light switches, it's that they used single-cell RNA sequencing, scRNA sequencing, instead of the traditional bulk RNA sequencing we've relied on for years.
And that technological leap, going from bulk to single-cell sequencing, it fundamentally changes what we can actually observe in human biology.
Okay, let me test an analogy on you to see if I have this right.
Let's hear it.
I think of bulk RNA sequencing like making a fruit smoothie.
Yeah, I like where this is going.
If you want to know what's in a fruit bowl, bulk sequencing is like taking the strawberries, the bananas, the blueberries, and maybe like a single rogue piece of kiwi and just tossing it all into a blender.
Right, just pureeing the whole thing.
Exactly. And when you take a sip, the banana completely overpowers the palate. You get the average taste of the whole bowl, but the subtle tartness of that one piece of kiwi is entirely lost.
Yep.
But single-cell RNA sequencing is like pulling out each individual piece of fruit from the bowl and tasting it one by one. You get the exact flavor profile of every single element.
That captures the essence of the technology perfectly, because the human lung is exactly like that fruit bowl.
It's a mix of everything.
Right. It is incredibly complex. It's made up of macrophages, alveolar type 2 cells, ciliated cells, and so many others.
So when these researchers used a pooled multiplexing strategy to sequence these cells individually, what did they actually find?
Well, they didn't just find a few neat connections. They identified 4,341 independent EQTLs acting as switches for 2,768 distinct genes across those 17 different cell types.
That is a staggering amount of data. But I have to admit, I'm a little stuck on why this is such a massive breakthrough right now.
Why is that?
Well, we have been doing bulk RNA sequencing on lung tissue for a very long time. Surely we had at least a fuzzy picture of these switches. I mean, a lung is a lung.
You would naturally assume that, right, that the strongest genetic signals would just show up in both methods.
Yeah, that makes sense.
But the data reveals an absolute shocker. Basically, 77.2% of these single-cell EQTLs are totally missed in bulk tissue datasets.
Wait, really?
Yes, they are completely invisible.
So over 3/4 of the genetic switches regulating lung cancer risk were just completely hidden from us this whole time.
Entirely hidden. Because in a bulk sample, just like your smoothie analogy, the sheer volume of the most common structural cells creates a massive amount of background noise.
The banana overpowering everything.
Exactly. That background noise completely drowns out the highly specific genetic switches happening in the rarer but vitally important cell types.
So if a cancer-triggering mutation only activates in a very specific rare immune cell, the bulk method just totally masks it.
It masks it completely. You'd never even know it was there.
Wow. Okay, so just to do a quick recap for everyone listening before we move on.
Good idea. There's a lot of information here.
So we define GWAs as the treasure map, right? And EQTLs are the light switches.
Yep.
And scRNA-seq is our high-def lens that lets us look at the individual cells instead of blending them into a smoothie. And by doing this, they found over 4,000 switches, improved that bulk RNA sequencing actually hides almost 80% of cell-specific signals.
Exactly. That's the foundation of the whole paper.
So think about your own body for a second, to everyone listening. How many subtle genetic signals are being masked right now in your own organs just because they only happen in a tiny fraction of your cells?
It's a pretty wild thing to think about.
It really is. So, okay, if we finally have this high-definition lens that lets us see every individual cell, the next logical question is, what is actually altering them?
Right, the environmental factors.
Yeah. How do outside behaviors, like smoking, actually reach down and interact with these microscopic genetic switches? The paper mentions they use something called a MMSE algorithm to figure this out.
They did, yeah.
And I understand they are looking at real-world factors like age and smoking, but mathematically, how on earth do you isolate the effect of a cigarette on one single type of white blood cell out of a sample of 500,000 cells?
Well, it required an unbelievable amount of computational power. MASC stands for Mixed Effects Modeling of Associations of Single Cells.
Okay.
What it essentially does is systematically strip away the noise. They fed this algorithm data on 19 real-world covariates.
Things like what?
Things like age, sex, body mass index, and, crucially, smoking history. The algorithm models the baseline of what a normal, perfectly healthy lung should look like.
Like a control group.
Right. And then it mathematically isolates how each specific factor physically alters the cellular neighborhood.
And the physical impact of smoking wasn't just like a slight deviation, was it?
Oh, no. It was profound. Smoking emerged in the data as the ultimate shapeshifter.
A shapeshifter. How so?
The algorithm proved that smoking alone explains 2.1% of the cross-sample variability in lung cell types.
Okay, 2.1% doesn't sound that big to a layman like me.
I know it might sound small, but in population genomics, it is absolutely massive. It proved that smoking powerfully alters the populations of monocytes, which are a type of white blood cell, and AT2 cells.
AT2 cells?
Yeah, alveolar type 2 epithelial cells. They secrete surfactant and basically act as the lung's internal repair crew. So smoking literally, physically changes the cellular real estate of the lung.
That's crazy. And within that shifting, smoking-damaged real estate, the genetic switches are unbelievably picky about where they operate, right?
Incredibly picky.
The researchers noted that over 60% of the identified EQTLs and 51% of the genes they regulate are exclusively active in just one single cell type.
Yes, and that specificity is what basically turns our old understanding of cancer genetics upside down.
Because we used to think a variant was just universally bad everywhere.
Exactly. But a genetic variant doesn't just universally turn a gene on or across your entire body or even your entire lung.
It's like a system of keys and locks.
Right. No, that's a good analogy.
Yeah. So if you inherit a specific genetic mutation for lung cancer, that mutation is your key. And you carry a copy of that key in every single cell of your body.
Right, it's in your DNA.
But that key only sits into a lock, meaning it only actually turns a gene on or off in one very specific room of the house.
Yes.
It might only fit the lock in an AT2 cell or only in a macrophage. But in the skin cells or the heart cells or even the other lung cells, that key is completely useless.
That is the absolute crux of it. And when you combine those two findings, that smoking physically alters how many of those specific rooms exist in your lung, and that your genetic risk keys only work in specific rooms.
The collision.
Exactly. You start to see the precise mechanics of how human behavior and inherited genetics collide to create cancer.
Okay, so let's pause and do another quick recap.
Okay.
We just learned that smoking physically alters the lung's cellular makeup, thanks to that MASC algorithm. And we learned that over 60% of these genetic switches are highly picky, acting like keys that only open locks in specific cells.
Perfect summary.
So, a question for you listening at home. If a genetic risk factor only turns on in one specific immune cell, how does that change the way we think about developing cancer drugs?
It changes everything, honestly.
It really does. Which brings us to the most critical part of this puzzle. We know the genetic switches are highly specific to certain cells. But how did the researchers definitively prove that these specific switches are the ones actually causing non-small cell lung cancer?
Right, because correlation isn't causation.
Exactly. They couldn't just guess. They used a really intense filtration process to, map the suspects.
They were ruthless in narrowing down the field. They actually started with 127 candidate genes that previous broader studies had loosely linked to lung cancer. But to prove true causality, they demanded 4 strict levels of clinical evidence. And the foundation of this was a statistical method called colocalization.
Let's pause on that mechanism for a second, because that's a heavy term. Colocalization basically means proving that the genetic variant causing the disease and the genetic variant controlling the gene's light switch are the exact same variant, right?
Yes, exactly. They used a shared probability score for this, requiring what's called a PP4 greater than 0.7.
Okay, so it mathematically eliminates coincidence.
Right. But colocalization alone wasn't enough for them. They went a step further to prove the gene could actually be read by the cell, using a technique called scRNA-seq.
Oh, man. Okay, I'm not even going to try to pronounce the full name of that assay. It's something about transpose-successful chromatin with high-throughput sequencing, right?
You got it. But the underlying concept is actually quite beautiful.
How so?
Well, think of your DNA as an enormous, densely packed encyclopedia. Every cell has the entire encyclopedia. But if a specific gene is packed away in a tightly closed volume, wrapped around these proteins called histones, it simply cannot be read or expressed by that cell.
It's like a book glued shut.
Exactly. What scRNA-seq does is measure chromatin accessibility. It physically proves that in a specific cell type, the DNA encyclopedia is cracked wide open to that exact page.
So they use these really strict criteria to filter those 127 suspects.
Right, they graded them.
So going through the levels, some genes were just level 4, right? They only had an open DNA book, but nothing else.
Yes.
Then level 3 had a loose EQTL association. Level 2 had a strong switch, but failed the strict colocalization map.
Right, they missed the PP4 cutoff.
But they kept filtering until they hit the absolute jackpot. Level 1 evidence.
It's a gold standard.
Yeah, just 28 genes that had a significant switch past the colocalization math and had an open DNA book in that specific cell.
Wow. And when the dust settled on those 28 gold standard genes, 2 major cellular players stood out as the primary drivers of inherited lung cancer risk.
Which ones?
AT2 cells, again, a lung's epithelial repair crew, and macrophages, which are the immune system's local security guards.
I really want to look closely at the specific cases from the paper here, because this is where all this abstract data suddenly becomes a very real, very scary biological mechanism.
It really does.
There's a genetic variant they identified called RS3618040.
Right. This variant acts as a switch for a gene called TP63, but what they proved, using all those levels of evidence we just talked about, is that this variant only turns down TP63 specifically in AT2 cells.
Which makes those repair cells highly vulnerable. And then there's the second case, which I found even more insidious.
The CSP8 one.
Yeah, a variant called RS6731171. This one lowers the expression of a gene called CSP8. Now, CSP8 is heavily involved in apoptosis, right? Which is programmed cell death and just overall immune function.
Yes, it's crucial for the immune system.
But this variant lowers CSP8 across immune cells, monocytes, T cells, naive B cells, and macrophages.
And if you think about the function of a macrophage, it is supposed to patrol the tissue, recognize damaged or rogue cells, and eliminate them before they become tumors.
So by suppressing, say, SP8, this genetic variant is essentially putting blinders on the immune system.
Exactly. What's fascinating here is that it's hindering the body's natural ability to fight off the cancer before it even starts.
Which makes me wonder about the tumor microenvironment itself. I mean, if you have an AT2 cell that is actively mutating because its TP63 gene is suppressed,
Yeah.
and sitting right next to it is a macrophage with a genetic blind spot.
You've just described the perfect storm.
It's terrifying.
It is. The data strongly suggests that lung cancer isn't just a rogue epithelial cell mutating in isolation. It is highly likely that these genetically compromised, blinded macrophages are directly interacting with the AT2 cell.
So they aren't just ignoring the cancer.
No, they are failing to clear the damage and potentially even releasing signals that force those AT2 cells to de-differentiate and mutate into a malignant state.
Wow.
It is a deeply toxic relationship between two entirely different cell types.
So time for our third recap. We just went through the 4 levels of evidence, proving that lung cancer is largely driven by genetic glitches in AT2 cells and macrophages.
Right.
We saw how TP63 in AT2 cells and CASP8 in immune cells get suppressed.
Which creates that perfect storm.
Exactly. So asking you listening again, if your immune cells have a genetic blind spot, are they actually helping the cancer grow instead of stopping it?
It's a really dark thought, but the data points that way.
It does. But as we dig deeper into how these genes behave, the paper introduces a phenomenon called pleiotropy, which honestly just bends the mind.
Oh, pleiotropy was incredibly widespread among these non-small cell lung cancer risk loci.
Can you explain what it actually is?
Sure. It occurs when a single genetic variant has different and sometimes completely opposite effects depending on the cell type it is operating in. The researchers found that 47% of established risk loci show this two-faced behavior.
I was trying to wrap my head around this, and I keep coming back to a corporate analogy.
Let's hear the analogy.
Imagine the CEO of a huge company sending out a single company-wide hand signal. It's the exact same signal going to everyone. But the sales department has been trained to interpret that signal as sell everything immediately, while the accounting department interprets that exact same signal as initiate a strict internal audit.
That's brilliant.
One signal, 2 totally contradictory reactions based entirely on the department receiving it.
The paper provides a stunning biological equivalent of that exact scenario.
The BPTF gene, right?
Yes, they looked at a genetic variant at the 17q24.2 locus. This variant regulates a gene called BPTF. Now, in the AT2 cells, this variant pushes BPTF expression down.
Okay.
But in the macrophages, that exact same variant pushes BPTF expression up.
You literally could never see that if you were looking at a bulk tissue smoothie.
Never. The upward expression and the downward expression would mathematically cancel each other out in the blender.
It would just look like baseline noise.
Exactly. It perfectly highlights why single-cell resolution isn't just like a nice upgrade. It is absolutely mandatory for understanding this disease.
And to push the boundaries even further, the researchers utilized TWAS, right? Transcriptome-wide association studies.
Yes.
TWAS is essentially a highly advanced predictive modeling tool, isn't it? It takes the genetic data, predicts the gene expression levels, and then associates that predicted expression directly with the lung cancer trait.
That's exactly right. And by running TWAS at the single-cell level, they didn't just confirm old suspects. They discovered 24 completely novel susceptibility genes that the scientific community simply didn't know about before.
24 brand new targets.
Yeah.
Which brings us to the ultimate so what of the entire paper.
The clinical application.
Right. This isn't just an academic exercise in mapping genes. This is about clinical payoff. It's about drug repurposing. Because once you know the exact gene and the exact cellular room where the lock is broken, you don't necessarily have to invent a new drug from scratch.
No, you don't.
You can literally look at the existing pharmacy shelf.
That is easily the most optimistic takeaway from this massive data set. Let's look at a gene called CLDN18.
Okay, what does that do?
It's a tight junction gene active in AT1 and AT2 cells, and they firmly mapped a mutation here to lung cancer risk.
But the amazing part is pharmaceutical companies already know about CLDN18, don't they?
They do. There is a drug called zolbetuximab that is currently in phase 3 clinical trials for gastric cancer entirely because it specifically targets the CLDN18 protein.
So by identifying that same specific target in lung epithelial cells, This research basically opens a massive, ready-made door for treating a specific subset of lung cancer patients.
It's incredible. And they didn't stop there. They validated another target, the DLX3 gene, at the 17q21.33 locus.
And they proved this one in the lab, right? Not just on a computer model.
Yes, they actually moved from algorithms to actual wet lab experiments. They ran luciferase assays and Western blots, and they physically proved that this specific genetic variant promotes rapid abnormal cell proliferation, specifically in AT2 cells.
So if a patient's lung cancer is being driven by this DLX3 mutation in their AT2 cells, what do?
Well, there is an existing clinically approved drug called ixazomib.
Ixazomib, okay.
It's currently used for other types of malignant tumors, but biochemically, it is a known receptor inhibitor for DLX3.
The implication of that is staggering.
It really is.
It means we could potentially repurpose a drug we already have right now to target this exact genetic profile in lung cancer.
It is the ultimate realization of precision medicine. Instead of hoping a broad chemotherapy drug hits the right target, we are mathematically matching the exact microscopic lock to an existing chemical key we already possess.
So let's do our final recap of this section. We saw how genetic variants can be two-faced, which is pleiotropy.
Like the BPTF gene going opposite ways.
Exactly. And advanced predictive mapping with TWAs found 24 new genes. And finally, zolbetuximab and ixazomib are sitting there as ready-made drug candidates.
It's huge.
So, audience question. Could the cure for certain types of lung cancer already be sitting in a pharmacy just waiting to be matched with the right genetic profile?
I think this paper strongly suggests the answer is yes.
It really is a profoundly optimistic place to arrive at after wading through so much dense genetic data. To bring this all together, this paper proves that lung cancer is not just a singular, monolithic dark cloud that descends on the human body.
No, it's much more nuanced than that.
It is a highly specific, microscopic neighborhood dispute. It is a single genetic variant in a patrolling immune cell or a damaged alveolar cell, triggering a microscopic butterfly effect that eventually compromises the entire organ.
And for you, listening to this, the reason this shift in understanding matters so deeply is that knowledge dictates treatment. We are finally moving away from the era of bulk medicine.
No more fruit smoothies.
Right. The future of oncology won't be a sledgehammer swing blindly at the chest, hoping it hits the cancer harder than it hits the healthy tissue.
Because of the sheer scale of this single-cell EQTL mapping, future treatments will be a targeted sniper rifle, calibrated precisely to your unique genetic and cellular makeup.
It changes everything we thought we knew about how disease forms.
It does. Which leaves me with one final kind of mind-bending thought for you to mull over as you go about your day. We've seen today how our inherited DNA dictates the behavior of individual cell types with breathtaking precision.
Yeah.
And we've seen how outside factors like smoking can physically rewrite the population of those very cells.
Yeah.
So what other microscopic battles are being won or lost in your body right now, entirely based on the environment you are exposing your unique genetic code to?
Wow. It certainly makes you look at every choice and every breath a little differently. Thank you for joining us on this deep dive into the source material.
We'll see you next time.